was born when my mother was 19. She had topped her high school science class, decided to be a music major, and after a year, met my father and married. I was her little baby project, and we had a splendid time. And then when I was about 4, she fell ill, and overnight, things changed. And over the next 15 years, I watched as she became a shell of herself. As a patient's relative, the frustration of having to deal with physicians who would only speak to a part of her problem, the anxiety and effort in trying to communicate when communication was so hard. It had the effect not of depressing, because children are resilient, but of engaging me, of showing me how important the brain is, how devastating diseases of the brain can be. So as I graduated high school, I knew I wanted to study the brain. I went into medical school because in India, where I was born and grew up, that was the obvious way for me to study the brain, and knew I wanted to either be a neurosurgeon or a neurologist. Now I know those are very different personalities. As I learned more, I said the breadth of neurology is what I want to do. I would have colleagues in medicine say, why neurology? There's so much more you can do if you're a cardiologist. And I said, but in my lifetime, that's going to change. We're going to get the answers. And indeed, the 1990s, when I went into internal medicine and neurology, was the decade of the brain. And we had some very exciting news. The kind of illness that takes away a person's personality, personhood, ability to communicate, ability to think and remember, what we call dementia, was just beginning to be understood. We were learning that there was a protein called amyloid that filled up the spaces between the brain cells, that there was a protein called tau that clumped and destroyed the brain cells, the neurons, destroyed the communication between them. And we were learning that there were some genes we had found in people with Down syndrome that also seemed to be explaining the common Alzheimer's disease dementia that was the most common cause of dementia. It was an exciting time. The way I had trained in neurology, we did not really study dementia. Nobody studied dementia. It was just considered a sort of disease of aging. It had the stigma. Nobody wanted to admit to it. So when my husband decided he wanted to come to the Framingham Heart Study and become a cardiovascular epidemiologist, and we had a 3-year-old, and he wouldn't go anywhere without his daughter, and she wouldn't go anywhere without her mom, I said, why don't I come to the US, take the exams, and learn about this disease called dementia? So I did a behavioral neurology fellowship with a Professor David Drachman, who was the person who had found what's called the cholinergic hypothesis of Alzheimer's, which said that this chemical called acetylcholine passing from neuron to neuron was what was decreasing in Alzheimer's. This fellowship time was everything I had hoped it would be, working with clinicians who provided the most holistic care. But the frustration was that we could care, we could show we cared, but we really couldn't do anything to prevent the disease or even to prevent the progression of disease. We were giving people medications that increased the levels of the acetylcholine, and that made them a wee bit better. It slowed down the slope of disease a little, but not much. We knew there was this protein called amyloid, and so in mice, we were finding ways using antibodies, passive immunization to reduce the amyloid. And in mice, when you took away the amyloid, they seemed to keep their memories. They seemed to sometimes even bring back memories. But when we tried to do the same in human beings, the amyloid in their brain went down, but the memories didn't come back. So there was this challenge that we were looking at what was obvious. We were dealing with what we saw under the lamppost, but perhaps what we were looking at was just a marker, a biomarker, and we needed to understand what was actually starting and causing this disease. And so at that time, when I finished this training, I went back to India, came back and did a residency again in the US so that I could be a neurologist working in the US. One of the exciting things that happened, this was in the mid-2000s, was that the human genome had been sequenced, and we were able to study variation in the whole human genome, what we called a genome-wide association study. Before that, we had some idea of biology, often based on cells or worms or mouse models, and we said, we look at candidate genes and see if they increase the risk of disease. And we did find 3 genes that increase the risk of Alzheimer's disease, something called amyloid precursor protein and presenilin. And these are still genes that cause about 5% of Alzheimer's disease, the ones that begin very early. But for 95% of the people, these don't provide an answer. So we said, if what we know is not providing the answer, let's look at everything else, right? It's like, let's look at the entire genome and assume any part of it could be responsible. It's like finding the killer in a butler's conference. So we did what's called these genome-wide association studies. But there are about 1 million places in the human genome where there could be a difference between one person and another. So if you're looking at 1 million points and trying to see if people with the disease differ from people without the disease, we need a very large sample of people. By this time, I was working with what's called the Framingham Heart Study. This is a study of around 15,000 people living in the small town of Framingham, Massachusetts, about 20 miles from Boston, where for the last 72 years, people have been coming every 2 years, telling us about themselves, giving blood samples. And so 15,000 people had their genetics examined, and we knew who had dementia and who had not, because this had been followed since 1975. But that's not big enough if you're making 1 million comparisons. And so we reached out to colleagues in different parts of the world who were similarly doing population-based cohort studies and formed what's called a charge consortium. It took us a whole day and to come up with that name. It's Cohorts for Heart and Aging Research in Genomic Epidemiology. And over the last 10 years, as leader of the neurology working group within this charge consortium, I've had the privilege of combining with about 80 different cohorts, and as of now, we are looking at about 500,000 people and looking across the genome. What are we finding? One of the early studies was that there was not just genes that control the amyloid pathway and the tau pathway. In fact, about 6 years ago, we did a big study and found some genes that looked very promising, TREM2, but they were not even expressed in neurons. We said, how could this be causing the disease if this protein is not even synthesized in neurons? And then we found that with colleagues who were working in the multiple sclerosis field, and that, by the way, was the disease my mother had, we found that these genes were expressed in microglia, which are sort of the immune police of the brain. And now we know that inflammation, immunity, microglia. These may provide drug targets for Alzheimer's. So what this genetic exploration without preconceived notions has achieved is point us to about 7 or 8 new pathways which are potential drug targets. Why is that exciting? Because another thing that's happened in the past decade is once we know a gene or a protein that's involved, that's abnormal to cause the disease, we are able to change that. We are able to either tune it down or up with molecules called antisense oligonucleotides. We are able to put a good form of the gene in a harmless virus and take it to that place where it needs to be expressed. We are able to use small molecules to change that gene's function. And so there is now new hope. Diseases like spinal muscular atrophy, where babies wouldn't live for their first birthday because their muscles were atrophying, are now treatable with these new gene-based approaches. Genes like sickle cell anemia and a kind of retinal disease, a kind of dementia called Huntington's disease, where we are now able to use this gene-based therapy. And so there is hope that this vast spectrum of people with what we now call Alzheimer's dementia, that this is a group with many subgroups where of the more than 40 genes we now know, and many others that we will find over the next 4 to 5 years, there will be some that are addressable. When I was a child, there was no treatment for multiple sclerosis. Today, my colleagues with the disease are practicing physicians. And I believe that in my lifetime, we'll be able to stop, perhaps bring back memories in this devastating disease that is dementia.